yesterday we just finished talking about the mitotic spindle. Tony, could you remind us of what that was, like the definition of what the mitotic spindle was?
Mitotic spindle?
Yes, that's what we left off talking about.
It's the, is it the, like, string that runs across, or the tubule that runs across the cell at the centrosome that pulls apart the?
Yes, correct. So it's these spindle microtubules along with the centrioles and these asters that come out. Could you tell me what the centrioles were?
The two things on the
Right, so it would be these two.
No, Emily,
but like a
did you have
definition.
a question too?
I did, but she answered.
Okay.
Oh, okay. So yeah, so like Tony said, basically, the mitotic spindle, what it's gonna do is you have these two centrioles that are on the two sides, and they're gonna shoot out these microtubule spindles, and what they're gonna do is they're gonna connect to these caricature molecules, and what they are is they're parts of like the chromosomal DNA, and they're gonna line in the middle with these microtubule spindles on either side of them, and then during anaphase, they're going, by the use of these microtubules, they're gonna separate into different sides. The spindle itself is going to disassemble because the strands are gonna depolymerize, and then the cell's gonna go through the process of cytokinesis. So that's just about where we left off yesterday. And so the next term that we need to know is karyokinesis. So basically what this is, it's like the division of the nucleus and its genetic material in there. And it comes right before like the division of the cytoplasm, which is also known as cytokinesis. And so the two daughter chromosomes, what they're gonna do is they're gonna split into two daughter nuclei. And like probably the most important thing to realize is that the genetic material is gonna split equally. So like this is the genetic material from here is gonna equally split into both daughter cells. And then that's gonna be followed by cytokinesis. And so cytokinesis is a little bit different in animal cells and in plant cells. So in animal cells, there's this thing called a cleavage furrow, and it's just kind of like, as you can see, it's kind of like a little indentation in between like the two cells that are like growing here. And so on like the cytoplasm side, what's gonna happen, there's like there's actin, microfilaments, and those are like the things that help us move. Those are like the muscles that help us move. And the protein myosin. And so the myosin and the actin filaments are gonna interact, and this is gonna cause the cell to contract. And through this contraction, what's gonna happen is this is gonna kind of push this, they're gonna kind of push the two cells apart, and two daughter cells are going to be formed through this process. And then in cytokinesis in plants, it's a little bit different. There's vesicles that come out of Golgi apparatus, and they're gonna line up in the middle of the cell. And so these plates are gonna keep growing until they fuse with the membrane of the plant, and this is gonna again form two daughter cells. So here you can see like the two vesicles from the Golgi move to the middle of the cell, and then you can see them getting bigger, and as soon as they fuse with the membrane, they are going to create two new cells.
Is there a name for that division between the two?
Between the two. I'm not sure.
The binary fission? Is that
No,
what that is?
for the plant cells, like what do we call that dividing line?
Cell plate. Is that what you're looking
Yeah,
for?
the
I think
metaphasic plate.
Yeah, I just.
Okay.
Alright. So yeah. Any questions?
I have a question on earlier. What's the difference between a caricature and a chromosome?
I believe I had it. Alright. Basically, I think it's like every sister chromatid has one. I don't know how to like. It is like here it is a picture. So you see like the when you go through the process, eventually you kind of see like the chromosomes they kind of split into two. So you have two of them that can go on separate sides. So the caricature, if I'm like understanding it correctly, it's like both of them together. I don't know if that makes sense. Like the two sides. It's like the two sides of the sister chromatid.
Doesn't the caricatures like help pull the chromosomes apart too?
The caricatures, they hold the material. Like what they say, part of the DNA, but it's like, like, part of it has to go to both sides,
Mm-hmm.
right? So the connector holds both sides of the DNA.
Yeah, I always like to think of it, I call it connector, I call it kinetochore, but, it's sort of like the glue that
Right.
attaches to the chromosomes so that the mitotic spindles can pull them apart.
Right.
So I think of the kinetochore as a way of holding onto the chromosome, sort of like gluing to it.
Right. Any other questions? Thanks for listening, guys.